Hello, this is part 2 of chapter 7, DNA structure and gene function. And we had just previously looked at transcription. And today we're going to continue that study and look at translation. So we have our processed messenger RNA that's ready for translation. If you need to go back and look at part 1 a little bit to refresh your memory, that's fine. Just watch the last couple minutes of the previous recording. So our processed messenger RNA is now a functional molecule. We've removed the introns, sewn the exons together, and it is ready to leave the nucleus and is moving onward to translation. So let's put that up out of the way and move on. So here's a quick review. If you've got the DNA template strand that consists of AGT, CTT, what would the nucleotide sequence of the complementary messenger RNA molecule produced in transcription be? So choose from any of these those options there. All right, well, I hope everybody got E. That would be the complement U to A, C is a complement to G, A is a complement to T, and et cetera, et cetera. All right, so now we're ready. Translation is the place that's going to produce the product of an actual protein. So how we integrate a ribosome into this process is what we're going to analyze right now. So messenger RNA carries the information for building the proteins that it got from DNA. It's going to go out into the cytoplasm, find a ribosome. Its cousin, transfer RNA, is going to help in the process of building this protein, and this whole thing is called translation. The building protein process involves the information that's carried in the nucleotide sequence that we are now going to refer to as codons. A codon is simply a sequence of 3 messenger RNA nucleotides. Each codon codes for a specific amino acid. 1 codon, 1 amino acid. The order of the codons in the messenger RNA transcript is what's going to dictate the order of the amino acids in the newly formed polypeptide chain. So one thing we're looking at here is we know that all cells have the same genetic code. It doesn't matter whether you're looking at my skin cell, my liver cell, my kidney cell, my brain cell, they all have the same genetic code. And so do the oak trees and the squirrels that are in them, and the cow and the dog. All the organisms are using the same genetic code that involves 4 nucleotides of DNA, the 4 nucleotides of messenger RNA, and the same 20 amino acids that exist on the planet. So we have a codon that's going to consist of a 3 nucleotide sequence, and it's going to encode 1 amino acid. And that's what they're showing, 3 letters, complements to the DNA. We're going to refer to this little portion as a codon, and that codon is going to code for 1 amino acid. Now, I can't emphasize this enough. The sequence of the codons in the messenger RNA and a ribosome is what directs the sequence of amino acids into the polypeptide. Where did the sequence of codons come from? DNA. So a nucleic acid sequence is going to be translated into a protein sequence. And here's a quick little 90-second video to watch translation.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons.
Oh. Oh, what happened? Sorry.
The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
Okay.
How is the information in the mature
Sorry. Okay, so let's dissect that video and look at all we know about translation. So there are 64 codons that are possible in making up the genetic code. So the genetic code is this work that was done once Watson and Crick figured out the function and the structure of DNA. So how is it that we know there's 64 codons? Well, we know there's 20 amino acids in the world. And we know there are 4 nucleotides. So we have 20 amino acids and we have 4 nucleotides. So mathematics came to play a role. If indeed we just used the 4 nucleotides, that's supposed to be a 1. Let me erase that. Let me make that better. Well, maybe I'll use text. Okay, so we have 4, and in mathematics, if you had 1 nucleotide, so the A, and if that A coded for 1 amino acid, we would get 4 nucleotides that would code for 4 amino acids. So 4 nucleotides codes for 4 amino acids. However, that wasn't good enough. There's 20 kinds of amino acids. So to explore a little further, if we took the 4 and we raised it to the 2nd power. So 4 raised to the 2nd power is 4 times 4, and that would give us 16 amino acids. And if indeed, let me go ahead and see if I can make that. If this is 4 raised to the 2nd power, that gives us 16 amino acids that we could code for. But if the same 4 nucleotides, say we used 3 of those, if you used 3 of those, Well, then that's 4 times 4 times 4. And that would give us 64 possible codes. And we would code for 64 amino acids. Well, there's not 64 amino acids, but we know we have found all 20 amino acids in living organisms. So only using 2 nucleotides in combination would get us 16 amino acids, and that would not be enough. But if we combined 3 nucleotides, that would calculate out to being able to code for 64 amino acids. So since we know there's not 64 amino acids, some amino acids are coded by more than 1 codon. And you're going to see that in the list here in a second. There also are, of those 64 amino acids, one of them is considered the start codon. So you always see this 3 nucleotide sequence, and that indicates the start of a gene. It will bring methionine as the amino acid to the beginning of any gene. And then we have these 3, UAA, UAG, and UGA, that are stop codons. And none of those 3 code for an amino acid. So of the 64, 4 of them are start and stop codons. So we have 60 left. And when we realized that we could put 3 of them together, that's what generated this thing that we call the genetic code. That shows all possible combinations of the 4 nucleotides and creating messenger RNA codons that will correspond to the amino acid. Now, this took a lot of time and a lot of research and effort, and it looks pretty simple to us, but it wasn't simple at all. I'll blow it up so that it looks a little bit bigger. If you put the 4 nucleotides, so mathematician was called in, how can we put these letters together so we know we don't have a duplicate? So the way you would do this is you take your first set of 4 nucleotides, UCAG. Using that exact same order, you make the same letters go across the top, UCAG. Then, because it's a triplet, we need a 3rd codon, we need a 3rd nucleotide, and those UCAG sequence here is the same as the first and second bases, and they're at the end of each of your individual original nucleotides. So that when you're trying to figure out a codon, so if, and these words here are your amino acids that these codons code for. So you notice leucine has 6 codes that codes for leucine. And you'll begin to understand why it's great to have 6 different codes that's coding for leucine. Here's 4 right here that code for proline, 4 for threonine, right? You see the valine, the alanine. You see a lot of duplicates, and there's a good reason for that. So these are the amino acids. We use 3 letters to indicate those as well. So PAG, LEU, LEU, isoleucine is ILE. All you have to do is look on the chart, find the 3 letters. You do not have to memorize the names of these amino acids. You can simply find the first 3 letters. You will notice, again, organic chemistry, you'll notice a similarity between the endings on the words. Leucine, isoleucine, phenylalanine, serine, proline, threonine, alanine, valine, asparagine, histidine, glutamine, tyrosine. The ene on the end is telling you it's an amino acid. Now there's some grandfathered words in here. Aspartic acid, this has obviously acid characteristics. The tryptophan, here's tryptophan. You associate that with turkey usually. These are all grandfathered in. They do have an official organic name, but tryptophan is acceptable, aspartic acid is acceptable, So you can use those terms, and it's acceptable by the scientific community. So you've got this code that you're going to use to be able to understand how translation is going to happen. So here's your DNA strand. You're going to separate the DNA strand to make the messenger RNA. And you can see, look at the first 3 letters, and you know that this bottom strand has got to be the template strand, because the complement to T is A. If the top strand were the template strand, the complement to A would be U, and this transcript would begin with U. So we know the bottom is the template strand. We make our messenger RNA transcript. We examine it by looking at codons and making triplets of each of those. Notice there's no spaces, there's no capital versus lowercase letters, there's no periods, commas, there's no mechanics to let us know about the triplets. So a lot of times I just draw a line so that I don't get mixed up, because it will just be a long list of nucleotides. Then the translation is going to happen when the amino acids are brought over to correspond with the codon in the messenger RNA by transfer RNA. So transfer RNA is a very important molecule. Transfer RNA delivers the amino acids to the ribosomes. They're the ones that are going to complement the messenger RNA. So these guys are cousins. Transfer RNA knows messenger RNA, and messenger RNA has the codon. Transfer RNA has what's called the anticodon. The anticodon is just like the puzzle piece that fits. So codon creates a shape, and the anticodon is the puzzle piece that fits in there. It's highly specific. So you have one anticodon for every codon, and that means we have 60 transfer RNA molecules, 61 transfer RNA molecules that have an anticodon on them that is complementary to the messenger RNA codon. There's a binding site for the amino acid on this molecule as well. So you have an anticodon on one end of the molecule and a binding site for the amino acid on the other. There's also ribosomal RNA. The little r stands for ribosome. These are what we would see in the cell, and we call them ribosomes. They are made in the nucleus, in the nucleolus, where ribosomes are made. There's proteins mixed in, and the ribosomal RNA is where the actual translation happens, and the amino acids are linked together to form the polypeptide chain. And you're going to see there are 2 rRNA subunits that make up each ribosome. There's a small and a large. So if you're looking at a transfer RNA molecule, remember, it is a protein. So that's why this thing squiggles and wiggles and has all these bends and turns, because there's attraction and repulsing forces that create this shape. On one end of the molecule is the anticodon that binds to the codon on messenger RNA. And on the opposite end of this molecule, there's a binding site that will grab the appropriate amino acid. So these transfer RNA molecules circulate in the cytoplasm. The amino acids are in the cytoplasm. Everything's available to this molecule, and the transfer RNA anticodon is what recognizes the codon on the messenger RNA, so that we're sure that the amino acid this guy's bringing is the appropriate amino acid. And we know that it is because of the sequence of the anticodon. So the transfer RNA binds to the messenger RNA codon at the anticodon and then drops off the amino acid. Each one of these transfer RNA molecules can only attach one specific amino acid. So they can't just grab any amino acid. This particular sequence, UAC, can only grab this particular amino acid. So it's highly specific. Now, everything that's happening in translation is going to happen at the ribosomes. Here is the small subunit, and this is considered the large subunit. And they're showing you those two parts where you're seeing the molecular structures twist and turn, and supposedly the small is on the top and the larger is this big lower part. Ribosomes help the three types of RNA interact with each other to build the protein. The ribosomes are essential for the location of translation. So here's the large subunit. And when the messenger RNA comes out of the nucleus, there's messages and enzymes all over the place. And a transfer RNA molecule is going to bind to this large subunit. The small subunit is where the actual RNA transcript is going to land. So it will sit on that. Now, there's 3 steps to translation, just like transcription: initiation, elongation, and termination. So initiation is where everything gets started. The messenger RNA acts as a template, right, to know which anticodons to bring the specific amino acid to the correct place. So the small ribosomal unit bound to the messenger RNA, the large one binds to the first transfer RNA. And when the anticodon pairs with the codon, that's when they're telling you that's the start codon. That's let's get it. Let's go after this, making this protein. So your anticodon here paired with the complementary codon that's on this light green or olive green stretch of the gene in the messenger RNA, and the transfer RNA would already have the appropriate amino acid that it's carrying, and that will be the first amino acid in the protein, this methionine, because this is your start codon. The second transfer RNA enters the ribosome next to the initiator, as showed in the video. The anticodon must match the codon, and so it brings the second amino acid, and it forms this incredibly unique bond, the only bond that's found in proteins. It is a peptide bond. It is a covalent bond. It's very strong, and it's made as the amino acids are brought to the ribosome. So the first transfer RNA leaves the ribosome, but the amino acid stays behind. So the methionine he was carrying, he leaves, because when the second guy shows up with this glycine, the first one hands the amino acid to the new guy, right? And we're moving from A site to the P site to the E site. So as the transfer RNA molecule moves to the E site, he's handing his amino acid to the new guy that just showed up in the A site. So you're going to link all these amino acids using peptide bonds. Talk to you about how it forms, just like we saw in the video. And when the ribosome reaches the stop codon, right at the end of the gene, here's where your release factor, this big blob, comes in, and it binds to the stop codon. There is no amino acid associated with the stop codon, just this release factor. And that means the end of our amino acid polypeptide chain that we're making. And it's going to detach from the messenger RNA, and all things being great and then everything's perfect, this polypeptide will fold into a functional protein. They do bring up a real interesting aspect of translation about how efficient. So this green ribbon through the center, all this is your messenger RNA, and it's showing you multiple ribosomes that are attached to this messenger RNA molecule. They can do this all at one time, and you can make multiple molecules of the protein all at once. So that's what they're doing. They've all attached onto this thing, and new polypeptides are being made. So this picture just summarizes what we learned about the mantra of biology. DNA makes protein. Here's your protein molecule. And we know, well, they're telling you this is the non-template strand, and this is the template strand. The template strand starts at 3', it starts with a T, and when you look at your messenger RNA, you can see the very first nucleotide is the complement to the T, not the A. So you can figure out which strand was the template if you can see your messenger RNA strand. We identify the list of codons on the strand, or the list of nucleotides. We recognize them into groups of 3 that we call codons, and each codon codes for one amino acid. One codon, one amino acid. One codon, one amino acid. So DNA makes protein. The folding is very important. The protein must fold correctly after it's translated to enable it to carry out its function. So as long as everything is going well, it will fold correctly. There will be attractions and twists and turns and contortions, and it will create the shape that is necessary to carry out the function. They can fold incorrectly if the DNA sequence was altered in any way. If the sequence has different nucleotides, then the encoded protein may have the wrong sequence of amino acids, and so it therefore wouldn't function properly. And they give you the example of cystic fibrosis. Cystic fibrosis is a deadly, deadly disease. Now, the very next section in the book, we've gone through transcription and translation, and we talked about finishing up and getting a functional protein. So now we're up to 7.6, and section 7.6 talks about how cells regulate gene expression. And one of the things that stands out that I put here is that producing RNA and proteins requires a whole lot of energy. So like they give you an example in the book, E. coli spends 90% of its ATP on gene expression. That's a whole lot of their ATP. It doesn't give much room for reproduction and repair, anything like that. So it's very energy costly. And so they're explaining to you that cells know to save energy by only producing needed proteins. So what I tell students in class is that cells are what are referred to as just-in-time manufacturers. So it's very similar to BMW. We don't have inventory. We do not like to build up a large cache of inventory because it uses so much energy. So we make the proteins when needed. Now, both prokaryotes and eukaryotes are going to regulate their gene expression, but they're going to do it in different ways. We're not studying how bacteria regulates gene expression. We're just going to focus on eukaryotes and gene expression. So I like the picture here. It was a picture of a chromosome, and you could see this long DNA strand, and then the sequence begins. And here is the portion that's the gene that's in pink, and then there's the end of the gene. So how in the world do we control gene expression? Let's look at a short little video.
Eukaryotes regulate gene expression using many mechanisms to control if a gene is expressed, the speed of expression, and how long it is expressed. Various regulatory proteins bind to DNA and affect the ability of RNA polymerase to bind to the promoter site and carry out transcription. Efficient binding of the RNA polymerase increases the rate of transcription initiation. Gene expression can also be controlled by altering intron removal and the splicing of exons to produce different mRNAs. The faster mRNA is processed, the greater the amount of gene product. Gene expression can also be regulated by controlling mRNA's access to or the efficiency of transport channels in the nuclear membrane. The passage of the transcript across the nuclear membrane is an active process that requires mRNA to be recognized by receptors lining the interior of the pores. Many enzymes degrade messenger RNA. Gene expression can be regulated by modulating the degree to which the messenger RNA is degraded. The translation process involves many different proteins and amino acids. The rate of gene expression can be regulated by controlling these factors. The availability or activity of any of the amino acids or proteins can alter the rate of protein synthesis and the amount of gene product.
Okay. All right, now, see if I can do this one right. Yes, so we're not focusing on prokaryotes. There's a little section A in your book talks about prokaryotes. We're starting at section B, where eukaryotes have multiple levels of gene regulation. So let me emphasize to you that here, the regulation of gene expression, just in the very beginning, talking about the availability of the DNA through transcription, is the major way transcription is regulated in eukaryotes. If we can stop it before even transcription can produce the transcript, well, that's a good way to control gene expression. And when we say gene expression is the outcome of the production of all these proteins. So if you have curly hair, your curly hair is a result of the interaction of multiple genes giving you the curly hair. If you're tall, it's the interaction of all the proteins that regulated how long your bones were going to get, how long your muscles were going to get. If you're able to jump high, all of that is gene expression. Whether you feel like you have a little nose and big round eyes, how you look, how you feel. If you're always a happy, funny person, that's all gene expression. So gene expression is the production of these proteins and how they're interacting together to produce who you are. So the regulation of these genes starts in the nucleus. This winding, when the DNA winds around these histone proteins and forms a nucleosome, they get wound again tighter and tighter and tighter. Getting the spinning and tightly wound appearance that you see in the X-shaped chromosome happens at one point in the cell's life, and it's near the end. But this, where you have the nucleosomes created and then some twisting, that can happen all along the DNA. And when it gets twisted or wrapped around these nucleosomes so tight, the enzymes cannot get in there to have access to the nucleotides. So that's why they're telling you some genes are wound up very tightly and cannot be used for transcription because they're just not available. But the other genes are. So that's the first regulation. There's many different proteins called transcription factors that can bind to the gene and affect the activity of that RNA polymerase enzyme that's going to bring the free nucleotides over to complement the DNA sequence. So these transcription factors can cause some problems in that if the correct transcription factor is present, the RNA polymerase reacts in a positive way when it finds the correct factors there, and it will transcribe. Another regulation. The transcription factors will bind to nucleotide sequences, and there's sequences again all along this DNA, and these particular sequences are called enhancers, and they enhance the transcription of the gene. So when you have transcription factors attach onto these enhancers, it activates the RNA polymerase. Whereas if they weren't there, the RNA polymerase wouldn't even begin transcription. And so the protein won't be made. So it's a way to control gene regulation. Okay, so you don't need to worry about the TATA box, the promoter. You don't need to worry about the folding and this whole clump of transition factors. It's a very complex process that they're just showing you pictures for, but you're not going to be responsible to know that. Now, some eukaryotic genes can encode multiple proteins. So we talked about the alternative splicing that existed between the introns and the exons that were cut out. The introns cut out of the transcript and sewn together. Well, by having alternative splicing and create a different gene from the same messenger RNA is another way of controlling what protein is going to be expressed. The fourth way they have is that we've got the messenger RNA and it's all processed and ready to leave. Well, it's got to go out these nuclear pores. There are plenty of proteins that are located at these pores that can interfere with the messenger RNA coming out of the pore. So restricting and keeping the messenger RNA inside the nucleus will certainly keep the protein from being created. And then the fifth method involves RNA degradation. And so you have messenger RNAs that come through the nuclear pore, but they're met with a whole bunch of enzymes, and those enzymes will degrade these things very quickly. And then there are another aspect, there are other aspects here that is, they're talking about these microRNA sequences that are going to bind to messenger RNA, and they prevent translation. So it's almost like an interference of sorts. These microRNA discoveries are fairly recent. They've been researched and discovered in the last 10 years, and it's a whole new science field that's opened up, and we don't know a whole lot about those microRNAs. So if you're interested in this type of molecular level research, that might be a field that would be of interest to you. And then finally, you have the actual protein that gets translated. So here's our protein, and we already made mention of the fact that they must be folded properly. And then they must be able to reach the correct cellular location to be able to carry out their function. So in that process, the cells can add and remove chemical modifications to the proteins that are going to change their activity. And also, there's chemicals they can add that will begin the process of degradation to that protein, so that this protein never gets to the location it needs to get to carry out its function. So I thought I had a little video after that. I thought it's not showing up as a link, or is it? Maybe it is. No. Okay, there we go. Here's the video. Okay.
Eukaryotes regulate gene expression
Oh.
using many mechanisms to control if a gene is expressed, the speed of expression, and how long it is expressed.
Okay, sorry, that is the one that we just watched. All right, so the very next section in the chapter now moves straight into mutations. So we've got our DNA transcribed, the transcript is moved into the cytoplasm, it was translated, our protein has been created. We've gotten through all of the regulatory mechanisms that ensured that our protein was going to get folded properly. And now we are going to look at some problems that can occur. And a mutation is a change in a cell's DNA sequence. So at some point within the DNA, and it's usually when DNA is getting copied, that mutations come to be. So we've got several varieties that we're going to examine. This is the picture from the book that they show you as a mutation. It's a pretty severe mutation because this is a face, this is the head of a fly, and here's his antennae that would normally be in place, all goes right. But this poor sap here, well, he had a mutation and it caused legs to grow where his antenna should be. So I would think that's a pretty severe mutation for this guy, and it may throw off his balance and everything and interfere with his ability to fly. So we've also looked at sickle cell anemia and cystic fibrosis. We know that there are mutations that cause disease. It can happen with a single base substitution. And this hemoglobin is the one that they referred to in the past that causes abnormally formed hemoglobin, which causes the blood cells to take on abnormal shapes. And you see them here, they look sickled. They're much skinnier and they're fold and causes blockages, right? These guys end up getting caught on each other and form blood clots where blood clots shouldn't be. And that's referred to as sickle cell disease. Now, there are mutations that change the DNA sequences that are not always harmful. When we say that word mutation, it just gives everyone a thought of something bad. Well, there are some that cause illnesses, but we need to keep in mind from a science perspective that mutations cause variation that indeed makes life interesting, but more importantly, evolution possible. So that's one of the big aspects to mutations. In science, we don't necessarily associate mutation with something bad. It can change one or a few base pairs, or it can affect a large portion of the chromosome. The fewer nucleotides that are involved, probably the less impactful the mutation can be. When a large portion of a chromosome has been changed, well, you can imagine that the mutation is going to be significant. So the very first mutation that they bring to your attention is called a substitution mutation. It's also referred to as a point mutation, and it's simply replacing one DNA nucleotide base for another. It is a substitution. Often, these types of mutations are referred to as silent, and they're silent if the mutated gene encodes the same protein as the original gene. Why can it be silent? Because one or more codon can encode for the same amino acid. So I'll ask you to flip back in your ebook to it's page 130 that has the genetic code. And remember, I brought to your attention that there are 6 different codes for leucine. So if indeed, for example, as I'd said earlier, if you have CUU. And that's going to code for leucine, right? It codes for leucine. But if there is a mutation that occurred and it changed one of your nucleotide bases, and so now your nucleotide base has changed to your codon gets to be CUC rather than CUU. Well, here's the beauty of having multiple codes for the same amino acid. So I can have a mutation where one of my nucleotide bases changes, but it's still going to code for the same amino acid. And so as long as it's coding for the same amino acid, it's a silent mutation. Nobody's going to know the difference. I've still got leucine as my amino acid, and so I'm going to get the exact same protein. Now, that's one example of a substitution. You can have substitution mutation where it changes the whole base triplet. And if you have the whole triplet changes, well, you probably are going to call for a different amino acid. It won't necessarily be a horrible thing. It just would depend on what amino acid is added to what polypeptide. If you substitute an amino acid and it drastically alters the protein shape, right, which would change its function, such as sickle cell, it is referred to as a missense mutation. And when you were doing your DNA activity, one of the activities was using hemoglobin in a normal gene and a hemoglobin that's been sickled. So one nitrogen base was changed. I can't remember whether it was supposed to be a U and it was changed to an A or vice versa. But when you look at sickle cell and you examine the gene, you realize it is a missense mutation, and one amino acid drastically changed the function of that protein because there was the wrong amino acid in there. And so it causes the shape to change, and that changes the shape of the red blood cell that it's inside of. There's also a nonsense mutation. And a nonsense mutation has the base triplet specifying a particular amino acid, and it changes into one that becomes a stop codon. So that's what happens in cystic fibrosis. So you've got the gene for cystic fibrosis, and we looked at cystic fibrosis back when we were doing cell transport. And we looked at the chlorine receptor in the cell membrane, and it doesn't allow for the chlorine to go through. It creates real thick mucus, and eventually the person is literally drowning in their own mucus in their lungs. So cystic fibrosis is indeed a drastic, horrifying disease. The whole base triplet became a stop codon, which means that you shorten your whole protein. If a stop codon would happen in the middle of translation, that means that however many amino acids are in place, that's where it stops, and it will not go on. So the cystic fibrosis, what the information here that I included, the 1,480 amino acids are in that typical gene that codes for the chlorine receptor. But if someone experiences cystic fibrosis, typically the gene experienced a nonsense mutation and a stop codon was inserted, and it has stopped the formation of that receptor. And it only has 493 amino acids because the protein cannot be made properly. That's a significant difference, missing almost 1,000 amino acids. So nonsense mutation has a stop codon in the middle. So let's look at an example here. So suppose you have this messenger RNA transcript, and the first letter is an A, but only a mutation happens, a substitution mutation happens, and the A is replaced with a U. So I want you to look at your genetic code, and that's on page 130, and tell me how many amino acids will be in this polypeptide chain if U replaces A. If this replaces A, your first codon would be UAA, and UAA is a stop codon. So there will be none. How many amino acids? None. This is a stop codon, and so therefore you won't have any amino acids laid down. Now, what kind of mutation would this be then? If UAA is a stop codon, AAA would actually code for asparagine or ASN. But now it's coding for a stop codon. So indeed, I hope everybody got it. This is a nonsense mutation. So in your book, there is a table, 7.2, and it's got all these types of mutations. So they use the analogy of codons being similar to words that we use in our sentences. And so they put the original sentence, if you will, that would be the original messenger transcript, messenger RNA transcript. The one big fly had one red eye. Okay. Now, a substitution to that would be to change the E to a different letter, and this one happens to be Q. So this codon will not be able to code for the proper amino acid, but one big fly had one red eye. There's still a good bit left. It is a missense with substituting one letter. It could cause the whole thing to not fold properly due to this, but the majority of the molecule looks like it would still be intact. And part of it may not fold correctly. The nonsense would be that FLY was changed to UAA, for example. So there was obviously a stop codon inserted. And so the whole rest of that transcript was lost, and you only have the one big. So that's a nonsense mutation, similar to what we just looked at. Okay. Now they give you some wording here that you'll see over and over. Wild type means the original nucleotide sequence. And when we get into genetics, you will see organisms referred to as wild type. And a substitution is just changed out nucleotides. And if you only have one codon that's altered, only one amino acid in the protein will be affected. So the here is demonstrating that just this protein, this amino acid in the protein will be affected. How much of an effect, we don't know at this point. Now, the other things that were on that list, there's some other mutations. And yes, we've already mentioned that we can have mutations that are silent, but we know, look at the fly, he's experiencing a mutation that is devastating. So we have base insertion and deletions, and these can be very, very serious. An insertion adds one or more nucleotides to a gene, and a deletion is just the opposite, that removes one or more nucleotides from a gene. Either type is referred to as a frame shift mutation. So if you have the words, okay? So if you go back and you look at the original sentence, the one big fly had one red eye. This is every 3 letters, it forms a word. So a codon. And what the line is saying here is a frame shift. Okay, so if indeed we try to change, if I change this letter O, if I took the letter O out, but the only thing is that I can only read 3 letters at a time, the O wouldn't be there, say it was deleted, okay? I would have to read the next 3 letters. So the next 3 letters would be NEB. Then the next 3 letters is IGF. Remember, there are no spaces or periods or capital letters to indicate that the transfer RNA molecule has to read only capital letters, or only little letters. Or if there's a letter gone here, you just ignore that. Well, this guy has to read in 3 letters at a time. So if the O is gone, then he would be reading the 3 letters like this. And these 3 letters are not going to translate into the same amino acids that the original ones did. So let's look at what else it says on the next page. So there. So nucleotides can be added or deleted if they are done as in 1 or 2 at a time. It will devastate the reading frame. But if you use multiples of 3, like a codon, you can add a whole entire codon, which may add an amino acid, and that effect may not be nearly as devastating. But the point is if you add by a number other than a multiple of 3, if you added 1, if you added 2, if you added 4, if you add 5, it will disrupt the codon reading frame, like I just showed you. So the sequence of amino acids will get altered, and of course that would be interpreted as a missense, or if a stop codon is created by the combination of 3 nucleotides in a way that wasn't expected, it could result with a nonsense mutation. Either occurrence results with a non-functional protein. Insertion and deletions are terrible mutations, and you typically get a non-functional. So here is an insertion. If they're showing you the example here where they inserted a whole entire codon with 3 nucleotides. So insertion of 1 nucleotide will change every codon after the insertion. But when it's done in groups of 3, like a codon, it did not change anything after or downstream from the mutation. Fly had 1 red eye, still is perfectly fine, and you would get the same amino acids. The 1 big, you'd have the same amino acids. The wet would insert an entirely new amino acid, and how that would impact the final polyprotein, polypeptide, is not as devastating as losing your reading frame. So here's an insertion that involves the frame shift where we insert some random letter. So big is still there, but only we inserted a letter. So now you've got the Q, and so QBI becomes the codon. And then you see you got the BI, well, then the G okay, we have to go up here to the original, not the insertion of a new codon. So the one big, all right, let's put the Q. We'll put the Q in here. So I'm transfer RNA. I can only read 3 letters. So I read these 3. I read these 3. There's QBI, GFL, YHA, DONERE, DEY. So you can see that's where these came from. When you insert a letter, the whole reading frame shifted. You couldn't read the 3 letters that make sense and are in place. You have to read the 3 letters, even if a nucleotide is inserted. And it was everything downstream will be altered. And the same thing with a deletion. If I were to delete, right? If I just deleted the B, say I delete the B, everything downstream is going to be altered by the reading frame. The 1, that's fine. But now I've got the IGF, LYH, ADO, right? The reading frame shifts. So that's what you're experiencing here. And they give you an example of a deletion where the whole codon is deleted. So the 1 big is fine. Fly is missing, had 1 red eye. So again, you're going to get this is a mutation. However, how that impacts the final polypeptide is not nearly as detrimental as when you have a reading frame shift. Those are drastically devastating. So here's another picture from your book. There's the original DNA sequence, and they're showing you the reading frame disrupted. So GAC, GAC, here's GAC, and here's GAC, except there is a thymine thrown in. For whatever reason, 1 base was inserted. So now everything downstream changes. You don't have GAC here any longer. You have TGA. And the whole shift happens. 2 bases added. You see everything downstream changes. But if you do the 3, you'll see that the reading frame is the same here. You just have a brand new amino acid that's going to be added. So let's look at that real quick in an animation.
The nucleotide sequence in DNA determines the nucleotide sequence in messenger RNA and, consequently, the sequence of amino acids in a protein. A mutation in the DNA can result in a change in the amino acid sequence of a protein. One type of mutation that can occur, either spontaneously or as a result of a mutagen, is the addition of 1 or more nucleotides during DNA replication. Because translation of a gene begins with a specific codon and proceeds 1 codon at a time, the addition of an extra nucleotide shifts the codons in the mRNA. This type of mutation is called a frameshift mutation. A frameshift affects all amino acids incorporated beyond the original site at which the addition occurred. If the new codon generated by the frameshift is a stop codon, the protein synthesized will be shortened and is often non-functional. Another type of frameshift mutation occurs when a nucleotide is deleted during DNA replication. Deletion of a nucleotide in DNA results in a change in the codons in messenger RNA from the point of the deletion onward. The deletion changes the sequence of amino acids inserted into the protein.
Okay, so that just shows you a little animation. There are 3 reasons that we've kind of narrowed down to explain the causes of mutations. Because of course, everybody's like, well, what's making this happen? What's going on? Well, they're spontaneous mutations that really don't seem to have any explanation. They typically originate when DNA is being replicated. There are mutagen exposures. Make sure you understand the word mutagen. Mutagen is a combination of words generates, mutations. So a mutagen generates mutations, and those mutagens can be anything from harmful chemicals or radiation, nuclear accidents. Chemicals and tobacco smoke clearly have been responsible for many, many mutations that occur. Water and air pollution, think of Flint, Michigan. So all of these things are interpreted as mutagens and can cause mutations in your DNA. And then finally, there is this thing called transposable elements, or more commonly called transposons. That's simply DNA sequences from one chromosome that will jump into another chromosome. And if it lands within the gene, it can disrupt, excuse me, disrupt the function. So those are just three real general ways that mutations can happen. There's two ways that that you may experience a mutation. One of them can be genetically, where it is passed through the germline. The germline means that it comes as a result of the mutation that arose in a sperm or an egg. So the parent may not experience the mutation, but the offspring will, because the mutation has occurred in the sperm or the egg's DNA. These are not nearly as common. Okay, we don't have as many mutations caused by germline mutations. Somatic mutations are where most mutations occur. So soma means body. Somatic cells are all the cells in your body. Any cell you can name, a nerve cell, a skin cell, a liver cell, a kidney cell, connective tissue, epithelium, any of that, those are all somatic cells. The only other type of cell in your body are called gametes, and those are your sperm and your eggs. Somatic mutations are typically the ones that occur most often. So how does that happen? You have, right, here's our normal DNA ladder. You've got the A to T, C to G combos here. They don't have the colors right. Your key is not right. But something will come in and break the hydrogen bonds, right? These hydrogen bonds were broken at this part of the DNA. And what happens is they fold over on themselves, and you can get what are called thymine dimers between two thymines here. Well, those are supposed to be guanines. But the nucleotides on the same strand will fold over, and there's attractions between the phosphates and the nitrogen bases to each other. And that can create mutations. So the very last section in this chapter refers to the fact that mutations are very, very important for evolution. And they create the genetic variability that we have on the planet. So mutations are not always harmful. And that's a difficult concept to wrap your head around. But it's true. Mutations create different versions of alleles. So you could have an allele that codes for seeds, like in this grapefruit here. I guess there is a seed in here, but think of the oranges and the grapes and many of our fruits, watermelon. that we now have that don't have seeds. They're called seedless. Well, they did that through a different version of the gene. It's called an allele. So there was a version of the gene that coded for seeds. So the gene would be called seeds. The two different alleles are yes seeds or no seeds. If we're looking at yourself, think of widow's peak. So widow's peak is your hairline across your forehead. And a widow's peak comes down in the middle and it forms like a little point. That's called a widow's peak, right above your nose in the middle of your forehead. If your hair comes down a little bit and forms a little tiny peak, that's called a widow's peak. So the gene is widow's peak. The alleles are, yes, you have a widow's peak, or no, you do not. Those are two different versions of the alleles of the one gene. They're two different alleles of one gene. So genetic variation is going to be important for evolution, so that as our climate is changing, our environment is changing, organisms need to be able to survive. So whether their coat needs to thicken up, or as we're warming, it needs to thin out, those guys have to be able to evolve and adapt. And that we're going to get that through mutations. These plant breeders, they induce mutations to create new varieties all the time. And we tend to like that, right? We want watermelons without any seeds. What was one of the newest things they had in the crop species? What are they talking about? Grapefruits, that's rice, cotton, oats, lettuce, begonias. All of those things have had mutations done to them. And if you've ever bought like an azalea for your front yard, they come up with colors of the flower that don't naturally occur in nature, or they double the bloom so that it just looks so much prettier, a double bloom than a single bloom. All kinds of things like that they've been doing with our crops and flowers and foodstuffs that's been going on for decades. So you just becoming familiar with that makes you aware that mutation is not a dirty word. It can be a very good thing. So that wraps up chapter 7, and chapter 8 will begin next, and watch for that. And thank you for listening.